Hello everyone, this is our Cancer Cell Bio Group project. We are
Anushna, Hannah,
Lizzy,
and Rachel. Our topic for today relates to a specific type of ligand that has a role in stopping cancer cell growth, transforming growth factor beta, or TGF-beta. Unfortunately, as all cancers do, the normal properties of TGF-beta are either lost or altered. This can be explained by the hallmarks of cancer, which describe what is required for a cell to become cancerous. In 2000, to compile the rules of tumorigenesis, Douglas Hanahan and Robert A. Weinberg proposed the original 6 hallmarks of cancer. Sustaining proliferative signaling, where tumor cells obtain their own growth signals and don't require signaling from outside the cell. The emerging hallmark, reprogramming energy metabolism, where cells shift their main energy metabolism to glycolysis to fuel cancer energetics. Resisting cell death, where apoptotic signals are altered or inactivated, stopping apoptosis from getting rid of potential cancerous cells. The emerging hallmark, genome instability and mutation, where a cell acquires mutations that give it an advantage over other cells. Inducing angiogenesis, where cancer cells need to supply themselves with blood vessels by changing signaling. Being capable of metastasis, where cells are able to detach from their tumor of origin and spread across the body. The emerging hallmark, tumor-promoting inflammation, where substances released during cellular inflammation may lead to mutagenesis, sustaining proliferative signaling, inducing angiogenesis, for example. Immortality, where tumor cells often have altered telomerase, which allows them to divide endlessly without going into senescence after a certain number of generations. And the emerging hallmark of avoiding death by immune-mediated systems, where cancer cells can avoid death signals from immune cells. And finally, evading growth suppressors, where a cancer cell avoids normal cellular anti-growth signals. TGF-beta is one such growth factor that cancer can't evade. For this video, we are going to focus on how dysfunctional TGF-beta activity is related to a cancerous cell evading growth suppressors. How is it related to this hallmark?
Transforming growth factor beta, or TGF-beta, is in the TGF family of polypeptides. These ligands are involved in various functions of the body. Some of these functions include cell growth inhibition, immunoregulation, extracellular matrix production, embryonic development, and angiogenesis. TGF-beta is released in response to a myriad of stimuli, reflecting the diversity of its roles. These stimuli include inflammation, injury and damage, hormonal signals, and cellular stress. TGF-beta acts upon a cell extracellularly by engaging with respective receptors on a cell's surface. TGF-beta binding sends information from the outside of a cell and initiates a signal cascade that will lead to changes in gene expression and cellular behavior. TGF-beta acts upon a cell's growth cycle before the restriction point in the G1 phase. The restriction point of the cell cycle is the point at which a cell will no longer depend on extracellular input and switch to intracellular signaling via CDKs. If a threshold amount of bound TGF-beta is reached, the TGF-beta receptor signals will upregulate CDK inhibitors such as P15, P21, and P27. This upregulation will halt the progression of the cell cycle at the restriction point and further prevent advancement into the DNA synthesis phase.
TGF-beta binds to receptors 1 and 2. Once bound to the receptors, TGF-beta triggers a downstream cascade of signaling pathways, either canonical or non-canonical. In canonical or a general fashion, TGF-beta phosphorylates SMAD proteins to activate and regulate gene expression. The non-canonical or alternative pathway involves the MAP kinase signaling pathway, which regulates processes such as proliferation, differentiation, and apoptosis. Crosstalk between these two paths is required for cancer metastasis. With this, TGF-beta regulates stroma cells in tumor microenvironment. Within this microenvironment, myofibroblasts, extracellular matrix, inflammatory cells, blood, vascular networks, and cancer-associated fibroblasts are found. CAFs are the main producers of TGF-beta, and patients shown with high TGF-beta are prone to metastasis and poor prognosis. In early stages of TGF-beta, it can be found to have tumor suppressive functions. However, later stages show that cancer cells are resistant to TGF-beta's inhibitory cells. Its signaling can then promote cancer cell invasion, metastasis, and resistance. This is done by loss of function mutations in the key components of the pathways. Being that TGF-beta triggers SMAD proteins, if lost or disabled, SMAD protein mutations can lead to many types of cancers. More specifically, TGF-beta acts as an immunosuppressant that can stimulate suppressive T regulatory cells and downregulate cytotoxic genes that would inhibit cytotoxicity of tumor cells, which is a commonality between cancers.
Previously, a scientist by the name of George Todaro proposed that rodent and feline tumored viruses are capable of causing cells to secrete a factor similar to the epidermal growth factor, or EGF. EGF is responsible for cellular proliferation. 3 papers led to the discovery of TGF-beta. One of these papers was authored by George Todaro and Joseph DeLarco, which found that then named sarcoma growth factors, or SGFs, were secreted from fibroblasts transformed by Maloney sarcoma virus. SGFs could bind to the EGF receptor, but were not considered the EGF ligand. SGFs were able to cause these cells to become anchorage independent as well. The other 2 papers, one authored by Moses et al. And the other authored by Anita Roberts and Michael Sporn, were able to characterize TGF-beta following the purification of SGF. Our highlight focuses on Roberts and Sporn, who collaborated with Todaro's lab to find that, following the purification of SGF, the following 2 factors are associated with EGF, one that bound to EGF to cause transformation, which is named TGF-alpha, where the other is simply needed to be present to cause the transformation. This ligand is known as TGF-beta. Later in 1985, the Sporn and Roberts lab observed that adding TGF-beta reduced cellular proliferation, but sometimes it was able to have the opposite effect and increase it. Roberts and Sporn found that TGF-beta could result in the decrease of cancer cells growing in soft agar when they supplied them with TGF-beta, from which, based on previous research by Moses et al., they decided that TGF-beta could inhibit growth of cancer cells. Most importantly, they found that TGF-beta can serve a dual role for either promoting or inhibiting anchorage-independent growth in MYC-transfected Fisher rat 3T3 fibroblasts. While it was able to inhibit colony formation of these cells, it actually promoted growth in the presence of platelet-derived growth factor, or PDGF, and also stopped PDGF's growth-promoting effects in monolayer culture.
Research in TGF-beta has risen greatly in the past few years, as the importance of its regulatory role in cancer cells in the tumor microenvironment has been realized. As TGF-beta signaling can be tumor-promoting, as seen in cancer cells, it has become a target for new therapies and treatments. The TGF-beta signaling pathway has been implicated in the pathogenesis of tumor metastasis in cervical cancers, colorectal cancers, breast cancers, and many other cancers through epithelial mesenchymal transition, or EMT, through its secretion of interleukin-like EMT inducer. Though TGF-beta has been implicated in the EMT for a few years, the specifics it plays in specific cancer types has become increasingly researched. Importantly, the role other proteins and/or genes, such as cell division cycles associated 7 in esophageal squamous cell carcinomas, may have on inducing or suppressing TGF-beta signaling that induces EMT. Additionally, a rising amount of evidence suggests that TGF-beta is the major regulator of behavior and composition of the immune cells within the tumor microenvironment. TGF-beta is then implicated in cancer cells' ability to avoid death by immune cells, which is an emerging hallmark of cancer.
Due to TGF-beta's role in cancer proliferation and survival, there have been treatments created and are being tested as targeted treatments to eliminate or slow the function of TGF-beta. The many types of treatments include introduction of antibodies, silencing of gene expression, and receptor traps. Antibodies such as frezonulab can be used to block the activation of TGF-beta to interfere with its binding and neutralize TGF-beta 1. Frezonulab is a human monoclonal antibody that neutralizes active TGF-beta and is used to minimize immune effector function. Gene expression silencing with antisense oligonucleotide silencers such as trabedersen, a glioma therapy, targets TGF-beta 2 expression in glioma, pancreatic carcinomas, and malignant melanomas. AONs exhibit exon skipping, and it uses this skipping in TGF-beta 1, which causes downregulation of receptor transcripts, which reduces TGF-beta activity. Ligand traps, such as soluble TGF-beta receptor 2, FC, that binds to the extracellular domain of TGF-beta, have been found to reduce tumor growth and metastasis. It has also been found to induce apoptosis in primary tumors. These traps are used to block the interaction between TGF-beta and TGF-beta receptor 2, resulting in inhibition of TGF-beta.
Now we have reached the end. Now, in conclusion, TGF-beta is an integral ligand that cancer cells use to evade growth suppressors and to even help them metastasize. While TGF-beta was originally thought to just influence the process of cell transformation by its discoverer Warren Roberts in collaboration with George Starr's lab, it is now known that TGF-beta plays many important roles in helping cancer survive when mutated or lost. Current research on TGF-beta and proteins and genes that influence it will help to understand the nuances and mechanisms with which it functions. In cancers, TGF-beta allows cells to grow and metastasize rather than stagnate and die, as would occur in the presence of normal TGF-beta. TGF-beta has become a target in recent research for cancer therapies and treatments, which could potentially help to slow or stop metastasis of cancers.